All right, guys, what's up? Welcome back. Today we are going to start talking about special senses. Um, I've broken it into three sections. The first section is going to cover taste and smell. I figure it's easier just to break down the huge lecture into three individual ones. Then the next one will cover vision, and the third one will cover the ear as far as hearing and equilibrium and balance. So this one's pretty short. Keep it short and sweet. There's not too much crazy anatomy, although there is obviously some structures you have to recognize and how they work and what they do. Um, but the entire chapter, if you will, um, focuses on the five special senses, right? From smell, taste, hearing, equilibrium and balance, and sight. So today we're going to talk about these two. And then, as I stated, we're going to have another lecture for sight, and then we'll have another lecture for hearing and balance, so we can break it apart. What are we going to be talking about? Um, First off, all of these special senses are going to detect some type of stimulus, right? When it comes to smell and taste, it is very important to recognize that they will detect chemicals, respectively. Um, and the smell would be detecting odorants or chemicals in the air. And respectively, the taste will be detecting chemicals in food. That makes good sense. Both of these will utilize chemoreceptors. These are specialized sensory receptors that detect chemicals. And we see that it is slightly different than when we look at the mechanical component of hearing imbalance. They're going to be detecting some type of movement, usually that we will get to as far as fluid moving back and forth in the inner ear and triggering certain types of mechanoreceptors. So, depending on the stimulus and what their specific purpose is, you're going to have receptors that work to detect that. And lastly, the sense of light for vision will clearly detect light energy, photons of light, and we'll talk about that in some detail. And these will specifically utilize what we call photoreceptors. Photoreceptors are highly specialized cells that can detect a photon of light and transmit that to action potentials. The key thing here is that whatever the stimulus is triggering the respective sensory receptor. That's going to depolarize. It's going to send a message to a particular area of the brain so we can interpret our senses. The specific cells are called olfactory receptor cells for smell. We call them ORCs. Gustatory receptor cells. Get rid of this for you here real quick. For taste, and we will talk about both of these today. We have the macula and the crista and the organ of Corti. Um, we will talk about those. Actually, those should be switched, but it's all good. We'll get to that. And then we have rods and cones, respectively, for vision. First thing, taste. This just highlights some of the general components you should recognize. This particular section's not too bad. I'm going to try to tell you what I want you to focus on as we go through. So, as we stated, olfaction and gustation are both chemical senses. They're going to detect chemical compounds, respectively, in different types of media, if you will. Some of the differences between these chemo detections. We have a lot of olfactory receptors, a lot of different types to allow us to detect a variety of different types of odorants, as opposed to taste, which we will talk about, are much more generalized, if you will. And we only have five, right? So we have one specifically designed to detect sweet, sour, bitter, salty, and umami. And we will talk about what specific chemicals stimulate those respective receptors. When we look at the tongue, we see that it's got a whole bunch of little bumps and stuff on it. Uh, these are called papillae. If you haven't, you will see this in lab and see what they actually look like, and we'll take a view as we move through this PowerPoint. There's different papillae located in different areas of the tongue. You should just be familiar with that. I don't expect you to be able to label the different ones. But inside, if you will, the crypts of the papilla, you can see here you have taste buds, right? So these chemicals will get inside those spaces, if you will. And we have a lot of taste buds. So what we're going to dive into is kind of what this looks like, the cells that make it up, how we detect a chemical, and how that chemical is sent to a respective area of the brain. So if we just zoomed in on this part right here, you could see on here it talks about the gustatory cell, the basal cell, that sensory component. We have these little microvilli, these hair cells, and that's what we're going to be showing, kind of the breakdown of this taste bud on this picture, right? So they have 3 types of cells. Important, right? You will see sticking off the ends of this little taste bud or these little microvilli. That is extremely important. And they project through what we call the taste pore, which is basically the opening of that taste bud. And first off, we have the gustatory cells. I'm going to try to highlight them for you a little bit. These are the gustatory cells. They're kind of pinkish on here. And you can see that they connect to those microvilli. Those are your receptor cells, right? Those are the things that are detecting the chemical. And you can see here that they directly connect to the sensory neurons that will send a message to a particular area of the brain. Man, the next type of cell, probably pick a different color so you can see it, are called supporting cells. The supporting cells are kind of peach-ish, located here. And they are, as exactly as they sound, provide support for those gustatory cells, and they're located in between. And lastly, we have the basal cells. They are stem cells. And when the gustatory cells need to be replaced, this is what does so. And they're kind of found here at the base of that taste bud. So 3 types of cells. Obviously, one of them is specialized in detecting the chemical and acts as the receptor. This is just a histology slide. You can kind of see the breakdown of that taste bud and the respective cells that we discussed. So when we talk about the depolarization of the receptor, if you will, or what triggers these specialized receptors, we have receptors that detect tastants, and we have 5 different types. A sweet receptor is designed to detect sugar, right? Anything that binds to that receptor, respectively, will be most likely a sugar type of compound, like glucose and fructose. We have sour receptors, which are designed to detect hydrogen ions, H+, aka acid, important. We have bitter receptors that can detect things like caffeine, potentially poisonous substances. We have salty receptors that respectively will detect salt, right? Na+ sodium is salt. And we have umami receptors that are designed to detect amino acids, especially or specifically glutamate in food, and give that kind of savory stimulus, if you will. So you should be familiar with each receptor and what it's used to detect, right? This is kind of what happens. I expect you to know the intricacies of the signal transduction inside, but I'll summarize it on the next cell. So basically, whatever it is binds. You have this chemical cascade, and it's going to send a message through a first order sensory neuron that will then go to the specific area of the brain called the primary gustatory cortex, right? Obviously, different chemicals, for example, here, right? That sour ligand, aka the H+, will bind, cause a chemical reaction, I should say signal transduction, and it will send respectively messages through those first order sensory neurons. So you don't have to know what happens inside, but you should be familiar with this, right? So chemicals will bind here, boom, to those little microvilli, right? Those respective sensory neurons will depolarize, send a message through these sensory nerve fibers. That tastant, if you will, is initiating this response, and then it passes through a first order sensory neuron. As we talked about with sensory neurons, you're going to always have a first order, a second order, and this will carry messages to whatever respective brain region it needs to go to. So where would these be going to? We have something called the primary gustatory area of the cerebral cortex, right? So we learned what parts we have of a taste pore, if you will, or a taste bud. Once those chemicals bind, signal transduction happens, they're going to send a message. So first it goes through the first order neurons, which we talked about, and then this will travel through three cranial nerves. The three you should be familiar with are the glossopharyngeal, the vagus, and the facial, right? These are three cranial nerves that are responsible for sending taste messages, right? Then you could see they synapse at the medulla, and then it's carried to the primary gustatory area of the cerebral cortex. Key thing, three cranial nerves, be familiar, right? And we can see here this insular lobe kind of sits underneath that frontal and temporal, and we'll see it pop up again when we talk about olfaction. So taste, pretty straightforward. Know your receptors, know the cells, know the pathways. And now we get to olfaction. A little bit more detailed. Not much. Vision and the ear are far more intricate. But when we talk about olfaction, I gave you some generalized highlights to kind of help you out. Obviously, I'll give you your study guide as well to help you focus. First, we got to figure out where this stuff is located, right? We talked about the tongue for taste. Here at the top of your nasal cavity, at the top, right? You can see we have these little projections sticking off the olfactory bulb, right? The olfactory bulb is a key structure we will be showing you pictures of and talking about. And then, obviously, once these receptors detect some type of odorants, these messages will be sent to certain areas of the brain, right? So that's kind of where we are. If you remember from A&P1, we had this cribriform plate, which had these little holes in it and stuff, and that's where those olfactory receptors are going to kind of stick down into. So when we inhale, right, air comes in, air is going to contain certain types of chemicals. Those chemicals will bind to those receptors, and we will talk about that pathway. So a couple key things to highlight. We have the olfactory epithelium, which you can see project down through that cribriform plate into the superior portion of the nasal cavity, right, by that superior nasal concha. We have the olfactory bulb, which we will talk about, kind of what's in there and how that message is sent. And then you have the olfactory tract, which is your olfactory cranial nerve, which is going to relay those messages back to particular areas of the brain. So far, pretty straightforward. Chemicals bind, messages get sent in, right? This is just showing you an anterior view of kind of where those are located in your nasal cavity. You can see clearly that they're at the top. Now let's dive into it. So we have olfactory epithelium, right? The olfactory receptor cells are specialized bipolar sensory neurons. I'm going to show you what they look like on this picture, and they are designed to I'll show you probably on the next slide, designed to bind and detect these particular odorants, right? So These olfactory receptor cells are these guys. You can see that they have these little arms sticking out here that are designed to, and I kind of color coded it, so like the purple ones, if you will, and the green ones are those olfactory receptor cells. The supporting cells will be the peach-ish ones, right? Hence why I kind of color it peach. Those are columnar epithelial cells, just providing some support in between the receptor cells. And you have the basal cells, kind of similar that we did for taste. And those are the blue ones. And those are the stem cells that are designed to replace those olfactory cells. And that's why they're sitting kind of more, not by where you would be binding chemicals, right? They're further in. These are those little holes of the cribriform plate we talked about, right? Obviously, that structure there is bone. And we're going to talk about how they bind and send messages inwards to the olfactory bulb, right? So the additional cell you can see in here are the olfactory glands. These are groups of cells. I mean, it's probably not the best color there. I'll do it in red so you can see it. That are secreting mucus. So we can have this kind of layer that allows the one for it to be moist, but two, those odorants to kind of bind to it, stick, and give these receptors a chance to detect them, right? So this is pretty important, right? Those olfactory glands produce this mucosal layer that allows for moistening of the surface and dissolving of those odorants to allow you to increase detection. See, we have a lot of olfactory receptor cells. We have hundreds of different types that combine various types of chemicals, and we are able to detect a absolute ton of different types of odors. We don't go into too much detail, but you kind of have, I'll mention it when we go through the slide, but certain smells can trigger certain emotional responses, certain memories and things like that, and that's because of the integration into obviously the limbic system, which we've talked about before. These are your first order sensory neurons. So they're bipolar. We talked about that, which means they have an end sticking out here and they have an end sticking out up here. One of the key factors here is this is our first order, right? This guy's going to go over here, but this is our first order. So obviously the first order sensory neurons are going to be the receptors that detect the stimulus. They're going to synapse up here in the olfactory bulb, and then there's going to be messages that are sent out through secondary or second order sensory neurons. This is showing you histology picture, kind of what those dudes look like with the cilia here sticking out. Obviously supportive cells to the sides. And clearly that would be covered with a nice layer of mucus, right? So what happens? Again, you don't have to stress so much about the specific signal transduction. But when we activate these receptors, right? So let's see here, right? Some type of odorant is binding to the receptor on those receptors, right? Whole bunch of stuff happens inside via cyclic AMP, etc. And the key thing here is it allows for depolarization of that neuron. So you have the neuron, a chemical binds, it will depolarize, aka send a message, respectively through the axon of that neuron. Then that action potential will be sent to mitral cells. So I like this here. This is kind of the big picture, right? Chemicals bind, cell depolarizes, sends a message up to the mitral cells, which we can see here. Try to do it without closing down the, there you go. These are our second order sensory neurons. They are located in the olfactory bulb, right? The bulb that's got the thing sticking off it. I'll show you where the olfactory bulb is located in 2 minutes. But this is the big picture, right? Odorants bind, cell depolarizes, message is sent through the first order sensory neurons, which are the olfactory receptor cells. They synapse, right? In that olfactory bulb, the first meets the second. Obviously, neurotransmitters will be released there. And then the second order sensory neurons, which are called mitral cells, transmit that message backwards. Not always backwards, right? And you can see that action. Look at it again. Chemical binds, depolarization occurs. First order synapses with second order. We can see that again here. And we have something called olfactory adaptation, right? Probably familiar with this, right? You walk into an area, it doesn't smell the best. Maybe it's a porta-john or something like that. But after a period of time, you kind of don't smell it anymore, right? And that's because adaptation for the olfaction happens pretty rapidly, right? Once we have chemicals binding in repetitive nature, the sensitivity of that depolarization decreases, right? So once something binds, that sensitivity is decreased, make room for other smells and things. And after about 1 minute of exposure to that consistent chemical, you basically have insensitivity, right? So you kind of don't smell it anymore and you're not really aware of it. And that's the take home point, right? So we call that olfactory adaptation. Typically, perfumes and stuff like that, when people are trying to smell different scents, you can use coffee beans to kind of displace all that odorant and kind of reset the palate, if you will. Cool. This is what we just talked about. Same thing, messages go up first order to the second order. And here we can see that those mitral cell axons, aka what's projecting backwards, will travel through the olfactory tract, right? This is basically the component of the olfactory nerve. And we know that a nerve is just a bundle of axons. So these messages will be sent back through the olfactory tract or nerve. So big picture here. I like this. It kind of summarizes the whole thing. You can see that synapse of the mitral cells and whatnot. The olfactory epithelium binds the airborne odorants. We have depolarization. We're going to synapse with that first to second order neuron. And then these messages will be sent back to the primary olfactory cortex in the brain to be interpreted as scent, right? So we've seen this picture 100 times. There you go. These things will send messages backwards. That's the green highlighted. That's showing you the olfactory tract. And here, you don't have to know every single part on here as far as the piriform cortex and stuff. But as these messages get sent inward, they will go to a variety of different places, right? So it's kind of a simplified diagram, if you will. Again, the odor information goes to the primary olfactory cortex. So number one, the cerebral cortex is an area where those messages will be sent. Some of them might go to other regions. This area of the cortex is located kind of at the base of the frontal lobe, kind of near that temporal lobe, kind of similar to what we saw before with the taste of smell. And as stated, these messages will also integrate through the limbic system, allowing us to connect emotional memories and, a whole bunch of other stuff that deals with the limbic system. So a significant factor is that these messages do not always go to the thalamus, right? This is the only sensory pathway that does not have to go through the thalamus. It can go directly to the cortex and the limbic system. And that's pretty significant, right? Most other sensory information goes to the thalamus. It's kind of a relay station that we talked about previously that will kind of send messages to the respective areas of the brain that it needs to get to. So olfaction is significant because it does not have to. Makes sense. Olfactory tract goes to the cerebral cortex and the limbic system. So, as I stated, this is a relatively short one. We covered two of the senses. The next ones won't be as simple. So we'll talk about vision specifically in the next lecture, and then we will talk about the ear in the third lecture. See you next time.